Good evening. Thank you for joining us for this first installment of the Tomorrow is Here lecture series. We believe the University of Arizona Health Sciences is uniquely positioned to reshape the future of healthcare and to address critical global health challenges through relentless curiosity, interprofessional collaboration, and the intentional inclusion of diverse perspectives and experiences. The Tomorrow is Here lecture series will share research, breakthroughs, and discoveries from the University of Arizona Health Sciences. I'm Anne-Marie Medina. I am the Director of Corporate and Community Relations here in Tucson for the University of Arizona Health Sciences. We respectfully acknowledge the University of Arizona is on the land and territories of indigenous peoples. Today, Arizona is home to 22 federally recognized tribes, with Tucson being home to the O'odham and the Yaqui. Committed to diversity and inclusion, the university strives to build sustainable relationships with sovereign Native nations and indigenous communities through education offerings, partnerships, and community service. Tonight's lecture, titled A New Era for Science, Creating New Defenses Against Disease After COVID-19, will be presented by Dr. Deepta Bhattacharya. After Dr. Bhattacharya's presentation, we will open the floor for questions, and then we invite you to stay for a short reception. Now it is my pleasure to introduce tonight's speaker. Dr. Bhattacharya received his PhD from the University of California, Berkeley, and then completed his postdoctoral fellowship at Stanford University. In 2008, he began his own lab at Washington University in St. Louis, first as an assistant professor, and then as a tenured associate professor. Dr. Bhattacharya returned in 2017 to his birthplace of Tucson to join the Department of Immunobiology at the College of Medicine, Tucson, where his lab continues to study both stem cells and antibody responses to infections and vaccines. Please join me in giving a warm welcome to Dr. Deepta Bhattacharya.
Okay, so thank you, Anne-Marie, for that nice introduction. And it is indeed my birthplace. It was sort of weird coming back here after all those years and having the memories of a 7-year-old and trying to, place that in the context of my adult views here now. So I'm looking forward to giving this talk in large part because I feel like a lot of the things that I've done in webinars and things that I've given for the university are almost more retrospectives and explaining how vaccines work and whatnot. And this is actually a little bit more forward-looking, which is, I think, fun, in the sense where I can sort of tell you a little bit what I'm thinking about as far as what comes next. And whether I'm right or wrong actually doesn't have any bearing, which is good. Hopefully I won't be held accountable for that. So let's talk about the pandemic. I think it's fair to say that a lot of things have gone not really ideally in the pandemic, and I think that by some measures and estimates, but the World Health Organization estimates that something like 15 million people have died as a result of COVID-19, which is really just a staggering number when we think about it. But what I would like to do actually is to sort of glean out some of these silver linings and not focus on some of the things that have not gone well, but actually recognize and pull out some of the things that actually have gone well. And I think, speak to some of the things that have been going on in the background when it comes to basic research and the applications of some of the technologies and fundamental discoveries that allowed us to rapidly adapt to this particular pandemic. So the timeline here this is something that I pulled from the Yale School of Medicine that goes through the first year of the pandemic. And I think it's important to start from the beginning here, which is, the first cases, as best we can estimate, occurred sometime in November of 2019, and there's a full-fledged epidemic in Wuhan, in China, in December of 2019. And from that original mysterious epidemic, we knew exactly what the virus was by the midway through January of 2020. And so I kind of want to emphasize just the remarkable nature of that initial very first step and how we were able to figure out what this thing was that was causing these cases in China, and then being able to take that information and move very quickly to the development of vaccines, incredibly effective vaccines, as well as some of the antiviral pills that are now starting to become more and more available as well. So again, focusing on some of the things that actually have gone right, I think gives us a little bit of a path forward to some of the lessons we can adopt looking forward, and hopefully not having another pandemic again in our lifetimes, but being much more prepared for it if we do. Okay, so let's talk a little bit about some of the underlying technologies and basic discoveries and things that actually allowed us to figure out what was actually causing this pandemic, this epidemic, at least at the time. So I want to go all the way back to the early 1980s and the conception of the Human Genome Project. And so this actually was the brainchild of many, but I think the initial conception started somewhere in the early 1980s or so. And I think at that point it was seen as just a massively, just almost ridiculously ambitious project, where there was intent to sequence every single base pair of the 3 billion base pairs that make up our genomes. And so the effort, I would say the actual effort began in earnest in around 1990 or so. And the technology at that point, I mean, I think that maybe I'm dating myself here, but I think the technology at that point was more or less what it was when I first started graduate school, where you're sequencing maybe about 100 to 300 base pairs at a time, okay? Now think about that. And we're trying to get to 3 billion base pairs, and every single one of these really torturous sequencing runs is only giving you 300 base pairs. And a lot of times the quality of those sequences was pretty unreliable. And so I think that, built into this timeline was some expectations that there are going to be some technological leaps. Whether that would actually happen or not, had a lot to do, is basically dictated whether or not this project would be finished in time. If it stayed at the pace of what the technology was around the early 1990s, there was really no chance that this project would be finished within the 15-year timeline that people had envisioned. But now, thankfully, technology did dramatically improve during that period of time, such that the average sequencing rate per year started to increase exponentially. And so, at the time, if you think about it from start to finish, the estimates are that the Human Genome Project, from start to finish took about 15 years and close to $3 billion, okay? Now, if we want to sequence an entire human genome, it takes about a week and costs about $1,000, right? So I want you to think about just the pace, the revolutionary pace of those technological discoveries. And one of the things that I often tell my students, the students in my lab and the students that I teach, is that a lot of times it's actually not the scientific goals or questions that are so much new, so much it is the technological capabilities to answer those questions. And I think this is a perfect example of that. So let's dig just a little bit deeper into what I mean then in terms of those technological leaps. The old school version of sequencing that I was telling you about is called Sanger sequencing, and actually something that Sanger won the Nobel Prize for, because it really did, completely revolutionize what we were able to do. But the way that it works is that you just take one little piece of DNA, you run it on as gel, and I can tell you that those gels are not easy to pour, and you hope that your sequencing run works with radioactive nucleotides, and then you sort of read them through one by one, and if you're lucky, you might get a couple hundred base pairs. Again, that's the pace that we are trying to get through 3 billion base pairs of the human genome sequence. But what happened is just like many aspects of human nature. I mean, it's actually the major push started to really increase based upon competition. And in this case, the early competition came from a man named Craig Venter. And he's a bit of a polarizing figure, but what he did is he just decided that the pace at which the Human Genome Project was going was just not tenable. There's no way that they were going to finish. And he was sort of frustrated that they weren't adopting some of the things that he suggested, which were shotgun sequencing. So instead of sequencing one little base pair at a time, one little piece of DNA at a time, you generate all of these different fragments of the DNA and you sequence them all together, and then you sort of computationally stitch them together just like you would a jigsaw puzzle. And so, that then initiated this competition between Craig Venter, the company that he started called Celera, and then the Human Genome Project, which all of a sudden, instead of just being bound by sort of the normal slow pace of federal government projects, now said, wait a second, if we don't get ourselves into gear, we're going to lose to Craig Venter, and that's just not acceptable. And so then the pace of sequences started to really increase. And then what happened is that the commercial potential of those technologies really started to jump. And so now, instead of actually just sequencing things even by the way that Celera was doing, now we have these things called next generation sequencing or massively parallel sequencing. And so the way that these things work is that you take these pieces of DNA and you tile millions of them onto these arrays, and you sequence them all together all at the same time. Which is, and then you have these computational frameworks to basically say, this is the sequence, you stitch them together, and again, now within the course of a week and $1,000, you can get an entire human genome out of it, Okay, so now when the Human Genome Project was going, and really for the first at least number of years after it completed, I think it was actually suffering from some criticism where people were saying, well, what did we really learn from this? What are the translational outcomes that have come of this? What diseases have we cured by understanding and knowing the human genome sequence? And I think maybe that's not quite the right way to think about it, but think about all of the outcomes that came as a result of that project. And one of them, undoubtedly, were these sequencing technologies. So from those sequencing technologies, then, we're able to sort of move forward and now bring it back into the era of COVID-19. And some of those technologies that people were criticizing for not necessarily having an immediate translational impact now very obviously did. And I'll take you through some of that. So basically, again, what Sanger, the original old school sequencing, would allow you to do is to fish for one thing at a time, whereas the next generation sequencing allows you to just grab all of those fish at once and then see what's actually in there. So basically what this sequencing technology does, and it's something that we do all the time, both in my lab and in core facilities here on campus, is move from someone who is sick with something, and you don't know what that something is, to now really within a couple of weeks knowing exactly what it was that's making these people sick, which is sort of remarkable. And so from the very first outbreaks that started probably early December of 2020 or so, to just a few weeks after that, two papers came out that reported the sequence of the virus that was the root cause of these outbreaks. So these are actually from Chinese researchers who published these sequences, actually at really quite great risk to themselves, because this was not something that necessarily China wanted to get out at that time. And so, I think it just speaks to the openness of scientists and really sort of almost a certain degree of altruism in making sure that these sequences get out there. So again, starting from something where the translational implications weren't super obvious to now moving forward into something where we knew exactly what the sequence of the virus was that was causing these epidemics. And of course, we knew that those local epidemics rapidly spread to a global pandemic very quickly. And so, in the course of infectious diseases, I think one of the most important things to do is to act early. And that, in fact, is what was used in many ways, using some of these sequences, both to understand what was going on globally, but also to use those sequences to develop vaccines, which I'll get to in just a little bit. So now, as we move forward from that sequencing technology into what we have right now, we have real-time monitoring of the variants of the virus that are circulating right now. And so this is just a little bit of a table. This is a table that I pulled from TGen, who assembles and analyzes a lot of the sequences that we pull from the state of Arizona. And so what we knew is that the original strain of SARS-CoV-2, which is the virus that causes COVID-19, was not really that diverse through most of 2020. But then towards the end of 2020 and now up and through now, what's happening is that the virus is mutating fairly quickly. And we're sort of subjected to sequential waves of these viruses. And the nice thing now with the sequencing technologies is we can know that. We can know that in real time. We can measure the cases. And Ryan Sprisler's core here sequences every single positive case. So every time any one of you provides one of these saline gargle samples and it happens to test positive, we know exactly what the variant was that actually caused that infection. So that then allows us to measure in real time what's happening not only on campus, but then also assist our colleagues as well to basically know what the variants are. So again, what we knew is that we started with the original SARS-CoV-2 through most of 2020. Then we got a wave of the Alpha variant that rapidly took over, then the Delta variant, and now, of course, what we're facing right now is the Omicron variant. And again, we can monitor how that virus is evolving in real time and be able to deal with it, understand how those vaccines and prior infection-induced immunity might be able to combat it and how much the virus might be evading some of those defenses. One of the other cool things that's actually emerged from this as well, not only from sequencing technology, but just sort of basic epidemiological monitoring, is wastewater. And I'll show you just a little anecdote of some of the things that actually happened early on campus from this. But, it turns out that the virus, you actually start shedding it through stool for days before you actually start getting symptomatic. And so you can get an early look as to what is about to hit your community by sampling the wastewater and seeing what the virus looks like in there. And now there are actually places that will actually sequence the wastewater as well so that you know what the variant is. And so that's becoming increasingly important because now we have all sorts of ways to test ourselves. We can go to Walgreens, we can get a rapid antigen test, and then we can see what's positive, but those aren't things that are necessarily being reported to public health agencies. And so what the wastewater allows us to do is to sort of monitor in an unbiased way the variants and the viruses and the waves that are about to hit us, and again, allows us to prepare. So again, one of the really cool things that has come from sort of modern molecular biology in terms of monitoring the COVID-19 pandemic. So this is actually, I think, a super cool story that I think shows the utility of this from just something that came out on campus. And really, wastewater monitoring was something that was led by Ian Pepper and Charles Gerba on campus. And this is actually a Twitter thread that I've pulled a screenshot of from Charles Fishman, who sometimes writes for The Atlantic. And so if you just read through some of this here, and essentially what he promoted initially some of the measures that we were getting ready on campus to get ready for in-person classes in the fall of 2020. And one of the things was actually getting this wastewater epidemiology going. And so what that allowed us to do was to sort of catch a couple of early cases before anyone had symptoms in a dorm, and then go into that dorm, and then before any of the people had actually even known that they were sick, figure out who they were, and then pull them into the isolation dorms that we had set up so that that little bit of those couple of cases didn't spread to a full-blown epidemic, an outbreak on campus, basically shutting us down before we even had a chance. And so those are some of the things that I think, University of Arizona is one of the real leaders on, and again, shows you how some of the early technology in modern molecular biology allows us to sort of deal with the pandemic. Okay, but I think, my expertise and interest really lies in more sort of the biomedical and translational interventions that, tools that have been brought to bear in large part because of the modern technologies and a lot of the basic work that's been going on behind the scenes. And so vaccines are forefront among them. That's what my lab studies, as Anne-Marie had mentioned. And so, I think I've been watching, the development of these vaccines with great interest in trying to understand how these things work. So from the initial sequence of that virus published by the Chinese researchers, we knew that this was what was called a coronavirus. And so there are other coronaviruses that circulate amongst the general population. They don't cause particularly severe symptoms. There's 4 of them that circulate. All of us have been exposed to them at some point or another, probably made for a fairly miserable cold, but it's not something that's going to kill you. So the SARS-CoV-2 that was causing COVID-19 was clearly a different beast than those earlier coronaviruses, but at the same time, it was very clearly a coronavirus. It's a kind of coronavirus that's made up of RNA. It's a little bit different than the DNA that makes up our genomes. And what happens is that when it infects a cell, it creates a couple of dozen different types of proteins or so. And those couple of dozen different types of proteins allows it to make more copies of itself, which are then released from the cell and then start to infect some of the neighboring cells around it. So that's how it causes disease. So the question is, okay, now we have this genome. How do we make a vaccine out of this? What is the real part of the virus that is its Achilles heel? What should we be aiming our immune response against? And so there's a couple of dozen different choices, and you don't really know which one. You could try all of them, but historically, those are the kinds of vaccines that haven't been that great. If you make immune responses against all different parts of the viral proteins, sometimes it actually can be a little bit distracting. And what you would really rather do is focus the response against the parts of the virus that really matter. So how do we know what matters? Well, we know this because of decades of basic immunological research on other coronaviruses. And so that starts from actually a mouse coronavirus called mouse hepatitis virus. It's actually a bit of a problem in some of our mouse colonies because once it gets in there, it can make the mice really sick. And so we do everything we can to try and keep this out of our mouse facilities. But then there have been a nice groups of people that have done basic research to understand the mouse immune response to this particular virus. And so I just want to point out here, this headline this title that I pulled here is from 1990. And there's actually even earlier work that was done on some of the common coronaviruses that date back into the mid 70s or so. And so this is, in some ways sort of thankless work. You could ask, well, why are you studying this? I mean, this is a mouse coronavirus. Why do you care? What disease does it cause? And I think hopefully, what the pandemic has taught us is that it taught us an awful lot in terms of preparedness. And so from these studies, what it turned out is that the immune response really needed to be aimed at this particular protein that the virus makes called spike. And I'll show you why it's called that in a second. That was shown both first in the mouse coronavirus, MHV. Then it was shown in the first SARS coronavirus. And that actually is a bit of an interesting story in and of itself, because it actually turns out that the first SARS coronavirus is far more lethal than the SARS coronavirus 2, which is causing the COVID-19 pandemic, in the sense that the fatality rate was something like 10%. It was extremely high. And so in some ways, I think this was a very scary sort of warning red flag moment for academia and for biotech to say, like, okay, we probably need to be ready for something like this. The first SARS coronavirus actually didn't take off globally simply because it didn't really have this period of pre-symptomatic transmission, so that you were really only transmitting to other people once you were already feeling pretty terrible. And so then that makes it a lot easier for people to isolate and quarantine and sort of use non-pharmaceutical interventions to try and shut the pandemic down. So that one, to the best of anyone's estimates, infected something like 8,000 people globally or something, as opposed to now what we know is that billions have been infected by SARS-CoV-2. And then finally, there is another virus that was also quite scary, had an even higher fatality rate, called MERS. And this is a coronavirus that infects camels. And in the Middle East, there were some outbreaks of this particular virus where the fatality rates were upwards of 20%. And so there was a lot of things where people got ready and understood the biology of these viruses, even though they never actually caused widespread pandemics. And so I think maybe that's one of the lessons that I'm going to keep coming back to, is that building that breadth of knowledge, even for viruses that aren't causing global pandemics, is actually really important. Okay, so the spike protein, what is it, and why is it called that? So this is some high resolution cryo-electron microscopy of the virus itself, which is, again, one of the real technological innovations here, is that we can basically zoom in and see what the culprit is, these tiny little viruses here. So this is basically, this viral circle here is 100 nanometers, just tiny, tiny. One of our cells is somewhere in the vicinity of 10 microns. So this virus is, the diameter of the virus is 100 times less than one of our cells. It's tiny. So this little guy here is the source of all of our problems in the COVID-19 pandemic. Now, what you can see here is that there are these little protrusions coming out of these circles. That's the spike protein that we're looking at there, okay? And so that's the part of the virus that it uses to get inside of cells and infect them. So let's take a slightly zoomed in look of what those spike proteins look like. And there's a few different ways you can get to this with higher resolution. So you can use that same cryo-electron microscopy technique. You can also use what's a classic technique called X-ray crystallography, where you make crystals of this protein, and then you use X-ray light to generate these diffraction patterns and say, okay, this is what that protein looks like. And so every protein has its own shape, and it's the shape of that protein that gives it its function. And so this is what the spike protein looks like. It's kind of an interesting looking molecule here. And the part of this that I really want to point out is this green bit right here, okay? So this is the part of the virus that it uses to latch onto your cells and get inside of it, okay? And so therefore, it's also the Achilles heel of the virus, where if you make an immune response against it, if you make antibodies that stick to that part, then it prevents the virus from ever infecting cells in the first place. So that's the goal of vaccination, okay? So that's a good thing. So it basically shows us where the vulnerability of the virus is. But there's a problem. This is a family of viruses that has evolved for, millennia, and it doesn't show this little green part for very long. And so this is what we call a metastable configuration of the virus, where it just flashes that little bit out just for the microsecond that it's trying to get inside of cells, and then it retracts that back down. So it doesn't give the immune system a particularly great target for making immune responses simply because it doesn't show it, okay? So then we have a little bit of a problem. How do we actually make good immune responses to a protein that does this? So this is, again, where some of the basic biology comes and some of the thankless work that preceded the development of these vaccines. So, good 10 years or so ago, the Vaccine Research Center at the NIH started doing some structural studies of other proteins from other viruses that behave sort of similarly to this particular spike protein, not knowing that this was going to come some point down in our future. And so Jason McClellan and Peter Kwong and Barney Graham started doing some of these interesting structural studies of another virus, in this case called respiratory syncytial virus, or RSV. This is actually a pretty big problem for kids, causes severe respiratory infections, and it actually makes a lot of kids really sick. And so it has a protein that looks in some ways very similar to the spike protein in the sense that the key part of it that it uses to get inside of cells, the virus doesn't show it for very long, okay, and then it pulls it back in. And so what Jason and Peter figured out was that you can actually modify that protein. Take a look at that structure and modify that protein so that little bit of the virus protein that sticks out and sticks to cells is actually stuck there, okay? So you just make a couple of little changes in some other parts of the protein, and then that part just gets stuck. It can't pull it back in. And so when they immunize with that engineered part, that engineered virus, then you get these incredibly good immune responses, things that you don't get just by if you didn't engineer the protein in the first place. So then they started to look at other viral proteins that were sort of like this, including one of the proteins from one of those common coronaviruses that just causes colds. Again, think about the criticisms that they get for this. Why are you studying this? This virus just causes a cold. This is a waste of time. You should be studying something that's already causing an epidemic. Those are the kinds of comments that they would get. But they just wanted to sort of make the proof in principle that the same kinds of changes that they made to the RSV protein would also work when they made it in a spike protein, even though it's a spike protein of a virus that doesn't cause particularly severe disease. And so what they figured out is that there's 2 mutations that they can engineer into this protein, then lock that little bit into place, and it gives the immune system a great target to go after, okay? So when they already had this, they had this prototype down already when SARS-CoV-2 came out, and they knew exactly what to do to engineer that spike protein into place, such that when the Moderna and the Pfizer vaccines came out, the immune responses you get from those vaccines are far, far better than what you get from a natural infection, okay? Which is, again, kind of remarkable. I just want to sort of map out the history of some of this. Okay, so I want to give this is a little bit of self-serving here, but I think this type of structure-based vaccinology really is the wave of the future. My own lab actually works on a family of viruses called Flaviviruses. So these include things like dengue virus, Zika, Japanese encephalitis virus, yellow fever, and the model system that we use the most is actually West Nile virus. And again, I will say that, again, West Nile has never been the source of a particularly massive outbreak, but hopefully some of those lessons that I've raised from the studies done by the VRC would also emphasize that it shouldn't just be the viruses that have already been causing pandemics, the ones that we should be studying. Okay, so this is what the cryo-electron microscope pictures look like of, this is now dengue virus here. And so it's actually a pretty beautiful structure here in terms that it forms these little lattices that allow the virus to do its thing. But it turns out that this particular family of viruses is a really challenging one to develop vaccines against because of a couple of traits here. So one is that there's a part of the virus where if your antibodies stick to it, then the virus can't infect cells. So that's good. However, there's another part of the virus where if your antibodies stick, it actually makes the infection worse. And that basically acts as a Trojan horse for the virus to get inside of your cells. And so what happens is that if you, for instance, are traveling overseas, you've been infected with dengue virus 1, that actually leaves you much more susceptible to severe disease later on if you get a dengue virus 2 infection that comes afterwards because of this Trojan horse effect, okay? And so for those reasons, people have been fairly reluctant to try and develop vaccines because of the worry that you might actually make it worse. So one of the things that structural biology then tells us is that we should be focusing the response to this part here and avoiding this part here. So some of the things that my lab does is now basically separating this out and then generating some of those same types of mutations that other groups have done for other viruses to make sure that the only thing you can aim your response at is the Achilles heel of the virus, so that some of these enhancing effects don't happen. Okay, but let me now return back to the mRNA vaccines and how they work and why they've been so successful. So we now know what exactly the mutations are in the spike protein that allow the immune response to get a good target and to aim its fire at the parts of the virus where you want it aiming its fire. Okay, so now that started from the viral sequence. We know what parts of the viral sequence to modify, and we know what the protein should look like that you want your immune system firing against. Okay, so how do you then go from that knowledge into developing an effective vaccine? So let me just start with a little bit of the central dogma here. And so the way that cells operate, normal cells operate, is that you have your genome that's in the middle part of the cell called the nucleus. And then it basically creates this template where you can copy little bits of it into a separate type of molecule called RNA. RNA is also the molecule type that comprises the SARS-CoV-2 genome as well. So the RNA then gets exported into a different part of the cell, and then that RNA gets translated into proteins that support the different functions of the cell. That's what's called the central dogma. That's how cells work. And so what the different types of vaccines do is, one way or another, by hook or by crook, tries to get the spike protein made so that the immune system can then generate responses against it. The immune system is generally geared to distinguish things that your cells normally make from things that really shouldn't be there. It's basically what's called self-non-self discrimination. And so what the vaccines, all they're really trying to do is to train your immune system against the spike protein so that it has a head start should you ever encounter the virus. So there's a few different ways and different vaccine platforms that were employed to try and make this spike protein. One is Johnson & Johnson and AstraZeneca, where they embed that spike gene into a harmless virus called an adenovirus that then gets inside of your cells and then instructs your cells to make that spike protein, okay, to give the immune system a target. The Novovax vaccine, on the other hand, is a little bit more complicated in the sense that it tries to make that protein initially and then immunize with that protein. And then the Moderna and the Pfizer vaccines are actually sort of, in some ways, taking a playbook from the virus itself and then using RNA to instruct your cells to make just that one protein, that spike protein. So that when you deliver this Moderna or Pfizer vaccine, the RNA is there and it instructs your cells to make the spike protein and then give your immune system a target to recognize. And so, what happens then is that there's actually some pretty big differences in terms of how easy or not it is to make these different types of vaccines. The Moderna and the Pfizer vaccine, for instance, is basically synthetic. You dial it up, you have your sequence, and then you just deliver it to your cells and you let the cells do the work, okay? And so for those reasons, the manufacturing of things like these mRNA vaccines has been much easier than some of the things like Novavax. And that's sort of been reflected by the vaccines that we have available to us here versus the ones that we don't. So the Novavax vaccine actually works pretty well. I mean, in some ways it works just as well as the mRNA vaccines, but it has been just brutal to get these things manufactured to scale and to have them meet the regulatory standards, to make sure that those proteins are pure, that we know exactly what it is that's being put into you. And Novavax has really struggled on that front. Whereas the Moderna and the Pfizer vaccines, again, you're letting their cells do all the work. And for those reasons, the timeline of those vaccines has been much, much more quick than some of the more traditional vaccine platforms or the ones built by Novavax. Okay, so now I'm going to go through actually just a little bit of a personal anecdote. And so maybe this is a bit of a disclosure too. So one of my very good friends is actually the founder of Moderna. And the history of this is that he had no intention of making vaccines. He's not an immunologist. He's actually a stem cell biologist. And so back in those early days, he just wanted to, get some different ways of delivering, different genes and turning genes on or off in stem cells so he could figure out how do you make them turn into this or that, okay? That was really the only goal. And so, but when he tried to start doing that, what he found is that the stem cells would pretty much explode and die immediately as soon as he tried to put that RNA into them. And he didn't know why. He says, well, that shouldn't be. I mean, cells make RNA all the time. So what is the problem? Why is it that when I put this stuff in them, it kills the cells? So then they actually forced him to learn a little bit of immunology and looked at some of the earlier work that were done by Kati Kariko and Drew Weissman at UPenn. And so what their early work showed is that RNA has to have certain modifications. You can't just take any old RNA, drop it into a cell, and expect that it'll behave like a normal cellular RNA. And the reason is that the cell does a lot of sort of chemical modifications of this RNA so that it knows this is something that I'm making versus something that a virus is making. So what Kariko and Weissman did is just in a, again, very thankless, decades-long projects, figured out what things can we do to the RNA so that the cell doesn't freak out and die. And so what they came up with is a series of modifications so that the intrinsic sensors of the cell that normally are looking for viral RNA no longer thought this stuff was viral RNA and made protein just as they should. So the unmodified RNAs that got put in the cell would basically kill the cells almost immediately, and they wouldn't make the protein, and there was no chance of an immune response that would come from that. But the modified RNA would actually allow the cells to make this spike protein and then draw on the immune system to make a productive immune response. So that's the basis of these vaccines. And again, it started from, at least from Moderna's standpoint, started from something that had nothing to do with vaccines. It was almost a little bit more opportunistic that they figured out that this was a good application. So if you're thinking about where the name Moderna came from, it came from modified RNA. That's where it came from. So that's Derek's wife, Nina, who came up with that name. So just a little bit of trivia in case you were wondering where that came from. Okay. So, and it turns out that this RNA isn't perfect though. It doesn't look exactly like a cellular RNA, and it triggers a little bit of inflammation. But that inflammation is actually really important to tell your immune system that there's something wrong, that this is something they should be responding to. And so it's almost like that early work, that sort of circuitous path that science sometimes takes that led us to figure out that this is actually the perfect solution to generate vaccines and immune responses to particularly emerging pathogens. Okay. So I think at this point, and probably most of the people in this audience know that the Moderna and the Pfizer mRNA vaccines were right up there with some of the best vaccines that have ever been made. And so both of them were reported somewhere in the vicinity of about 95% efficacy in preventing symptomatic COVID-19. And, when these data first came out towards the end of 2020, I mean, I've been doing this a long time. I've been a scientist for a long time. And I still don't think that I've ever been quite so excited or seen such beautiful data as this. And I probably will never see it again. These are the kinds of curves that you just don't anticipate. And so really what we're looking at here is the groups of people that received the Mrna vaccine. These are the people that received the placebo. And if there's really no doubt in terms of the incidence of people who got COVID-19, right? There's no question. You can look at this and you don't have to spend a ton of time explaining it. So that was a good day. And I think that we were sort of hoping that the pandemic would be put behind us at that point. But obviously what we know is that things have been changing, and mainly it's the virus that's been changing. And so, what took over in the summer of last summer, 2021, was the Delta variant. And now what we're facing is the Omicron variant. And so for those of you who aren't used to looking at some of these pictures, again, this is that famous spike protein that we were talking about, and that little bit of the Achilles heel that's exposed by the virus here. And so what happens is that this virus, when it makes more copies of itself, starts to make some mistakes. Most of those mistakes are deleterious. I mean, it basically borks the virus so that it, does worse. But simply by chance, some of them will actually improve the fitness of the virus. And then there's this selection process for the sort of viruses that actually have improved what they're trying to do, which is replicate, transmit to other people, and to evade the immune response. And so, what these differences mean, what these annotations mean, is that this is one part of the virus that has changed. The proteins are composed of 20 amino acids designated by single letter codes. And so this one then used to be a threonine and has changed to a lysine here. And so some of these mutations actually have some pretty important functional consequences. So for instance, this one down here in the Delta variant makes a big difference in terms of how quickly the virus replicates and transmissibility. And so, you can see that it doesn't have a ton of differences from what it originally started at, but this one makes a huge difference. And what it means then is you're exposed to much higher levels of the virus, and it's harder for the vaccines to deal with it. This one still blows my mind if you just look at the number of mutations in this particular variant here. It's just littered with red. And so the major ones, the problems of Omicron, are actually all of these mutations here sort of collectively lead to the evasion of vaccine and prior infection-induced immunity. And it's one of the reasons why the vaccines aren't working quite as well as they did from that beautiful Kaplan-Meier curve that I showed a little bit earlier. So now what I'd like to do is shift to what we're doing going forward to maybe deal with some of these problems. And then as we start to take a more 50,000-foot view, what some of the lessons we've learned from the vaccine might mean as we go forward to other types of infectious diseases. So again, what we're looking at here are these large waves that keep hitting us once every 6 months or so. And I think this is not really the future that any of us really hoped or wanted. And now the question is, what can we do about it? Can we at least make it so that it's once a year and it becomes a little bit more like flu? We know when to get our vaccines and whatnot. And so I think, at the minimum, that's the goal, but I think we should actually start thinking a little more ambitiously than that as well. And I think one of the ways that we can start to get there are what we call variant-proof vaccines, or what are called pan-coronavirus vaccines. And it sounds like it's sci-fi, but I think at this point, some of the literature that I'm seeing and the data that I'm seeing coming out right now actually start to make me think that it's really possible. And I tend to be a little more skeptical than most when it comes to these sorts of technological advances. And so the basic principle of this is that you take spike proteins not only from the original variant that was circulating, but then you basically put together these mosaics where you have all sorts of different spike proteins from all sorts of different viruses. And some of the more innovative ones actually include spike proteins from bat coronaviruses that haven't even reached us yet. And so then what you do is you create this huge diversity of these spike proteins to which you draw an immune response against. And in really some of the nicer examples of these kinds of strategies, you really can't find one of these types of coronaviruses that is capable of escaping immunity. And so some of these things now, I think, again, show you what starting from first principles allows you to do, because you can then estimate and figure out how exactly is it that these viruses are evading immunity, and can we build these mosaics that prevent that really from happening? So this is an example from Pam Bjorkman's group at Caltech here, and they've done these experiments not only in mice now, but also in non-human primates, and it looks amazing. So I think this is one of the futures of what we're looking for here. So there's a few different technologies that vaccine platforms that have adopted this technology. One of them is actually mRNA vaccines, where now instead of just having it produce one type of spike protein, you're actually having the cells produce a number of different spike proteins that are all assembled into these little nanoparticles. And your immune system really likes attacking things like that because it kind of looks like a virus. There are other ones that are just protein-based, and then there's actually a couple of them right now. This one here is actually in phase 3 clinical trials right now that's initiated by Walter Reed. I'm not sure that this would be my front runner, but ultimately what we've seen is that sometimes it's actually who's first more than who's best. But I think, when you add up all of these different things and these different results, I genuinely think this is possible, where we're not having to sort of play this whack-a-mole game with the different variants that keep popping up. I think one of the other things that we've learned from this pandemic, particularly in the US, is that it's not just vaccines, but vaccinations and who actually decides to get them. So here in the US, I believe it's only about 2/3 of the population has actually gotten the vaccine, and only about 1/3 has gotten the booster, which we think is important for providing at least some protection against Omicron. And so I think we have to sort of acknowledge the reality of the culture that we're in, obviously try to do everything we can to combat disinformation, but also recognize that the reality is that people are a lot more willing to accept cures than they are preventives, even though really in the context of infectious disease, an ounce of cure is worth many, many tons of cure, but the simple reality is that some people just won't get them until they're already sick. So here again, I think is another victory of doing some of that hard and thankless work on viruses that didn't necessarily become pandemics or epidemics. So one of those is Paxlovit. This is Pfizer's antiviral drug, which actually is remarkably effective at keeping people out of the hospital once they've already tested positive. And I think this is one of the real game changers when we're trying to think about what it takes to get back to normal. So this works on people who have, compromised immune systems. This works on people even after they've tested positive. And it just is just sort of, I think, really one of the, real game changers when it comes to, how do we actually get back to normal? But the story behind this is actually pretty interesting because Paxlovid was really developed on SARS-1. Again, the one that had 8,000 cases worldwide that never turned into a real pandemic. But the Pfizer scientists decided that they wanted to continue to pursue this drug and develop this really nice small molecule inhibitor of one of the key proteins of the virus. And so that it was more or less ready to go when SARS-CoV-2 hit. So this is a quote that I pulled from Derek Lowe, who writes a blog for Science magazine called In the Pipeline. And so I'll just take a second to read it here. So when he's talking about drug development, which is really his forte, he said this was really only possible because of that earlier work on SARS-1. And remember, that work looked like wasted effort. Again, they got so much criticism, both academic researchers and pharmaceutical companies got a lot of flack for studying things that weren't the cause of a pandemic. So there's no SARS to treat. There were no other related coronaviruses that were causing much trouble. But that's science. Once you discover something, it stays discovered. And that knowledge is out there waiting to be put to new use. So I think that has to be one of the lessons now going forward. So let's now bring it all the way back again to some of the things that did work. And I think one of the things that worked well was Operation Warp Speed. And I'm going to use the principle of what that slide I just showed you here to what I think we should be thinking about going forward. So this is the normal timeline of vaccine development. And basically what we're showing you here, there's 3 phases of vaccine development. There's the early phase where you just take a handful of people, you try to figure out the dose that's safe, that gives you a good immune response and isn't causing too much trouble in the people. You expand that group just a little bit just to make sure that dose you chose isn't causing some more rare side effects in groups of people. And then you finally move on to the phase 3 trial where you actually try to see if that vaccine prevents the disease you're trying to prevent. And so the reality is that this process normally takes years and years and years. But it's not actually the trials themselves that take all that time. It's all that stuff in between. There's usually decades of research to understand those viruses in the first place. Then the companies have to decide whether or not they actually want to move forward between each of these different phases, and then they have to figure out how to make enough doses of those vaccines, and then finally move to the phase 3, and even then understand whether it's worth pursuing that particular vaccine or not. So what Operation Warp Speed did, well, actually the first thing is, as I mentioned, there was a lot of basic work done on those coronaviruses that preceded all of this. So that really shrunk that timeline. We already knew what to target in this particular virus because of that basic work. Then what the government did is basically de-risked these processes and pre-committed to buying a lot of these doses so that there was really no need to have those delays in between these different phases. And the manufacturers were basically able to move straight from one phase to the next, start manufacturing and be ready for this critical phase 3 trial. Now, it actually turned out fortuitously that at the time that Pfizer and Moderna were testing their vaccines, the pandemic was raging in the US. And so it didn't take very long to accumulate a lot of infections in that placebo group and have that beautiful Kaplan-Meier curve showing how well those vaccines were working. So that was a little bit of luck. But at the same time then, it also allowed these companies to start manufacturing these doses even before the phase 3 trial had been completed. So there's some lessons to be learned now as we think about preparedness for other pandemics. I was at a conference at the American Association of Immunologists last year, and they brought in Tony Fauci to speak. And he was refreshingly very blunt with his assessment of the pandemic response, and that a lot of the public health responses left something to be desired. But really, one of the shining aspects of this was the immunology that led to the vaccines. And so one of the things he was really advocating for was being ready and acknowledging that we don't really know what's going to come next. And so what that means is studying families of viruses and then getting their vaccines and antiviral drugs through the phase 1 and 2 trials so that we can move very quickly should a new pandemic hit. So what would that then involve? So that means that we need to be thinking about and conducting, and the government needs to be funding basic research on families of not just viruses, but also bacteria and fungi. And I think that, again, building up that broad knowledge base and technology that allows us to move quickly. We also need governmental investment that allows us to move through these phase 1 and 2 trials, because again, left to their own devices, companies might make business decisions that aren't necessarily in the best view of a best sort of public good. And we need some governmental investment to make sure that we get these drugs through these. And then also have manufacturing and stockpiles ready to run those phase 3 trials should a pandemic hit based upon one of these families of viruses in which we've learned some of the basic biology. And if Tony Fauci is talking about this, I think this is actually where the governmental response is going to go, and it should leave us in much better position if the next pandemic hits. Okay, so genomic surveillance, I think, is actually one of the other important things here. Is that here we went from an undiagnosed set of infections in December of 2019 to knowing exactly what was causing those infections just a few weeks later. And so it sort of makes me wonder, why aren't we doing this for all infections? And our colleagues at Campus Health will tell us that most respiratory infections, we have no idea what's causing them. We've had a lot of kids showing up to Campus Health with respiratory infections thinking that it's COVID, and most of the time it's not. And so the question is, what is that? Can we learn something from that? And can we use some of these lessons from COVID-19 to develop some better therapeutics and some better vaccines that allow us to deal with things that maybe are just nuisances now, but could turn into something more problematic? So, for example, I'd mentioned that Ryan Sprisler's facility sequences every positive test that we get, but they also have the capability of sequencing anything that comes our way from an undiagnosed respiratory infection. And so from that information, one could imagine that you could take those same steps that you did for SARS coronavirus 2 and start to move it through the pipeline. So we start from an undiagnosed infection to knowing what viral family it's from. We grow up that virus. We follow that virus through some of the epidemiological measures that are now pretty standard across the country, knowing when those things are actually starting to become a bigger problem. We can then develop vaccines that are based upon some of the basic knowledge that we develop from those viruses and have those ready. So some of these things that we accept as just par for the course don't have to be. And then use some of those, again, informations to start developing antivirals against some of these viruses that, again, maybe just cause colds right now, but could turn into something a little bit worse later. And again, I think one of the questions is, well, that sounds like an awful lot of work, and does it make actually business sense to do so? I'll just leave it with this last slide here. And the cough and cold medications have earned something like $11.5 billion a year. And so, what if those things actually worked? Wouldn't that be something? And I think, again, the way that we consider infectious diseases, I hope has actually changed for the better when we think about all the lessons we've learned from COVID-19. So what are those lessons? So in my view, the basics matter. The basic and fundamental studies really broaden our knowledge and technology base. And so when people say that it was just the vaccines were developed in record time, they really weren't. It's built on the backs of decades of research that allowed us to move quickly. I think it's also important now to not just focus on the immediately translatable problems, because again, a lot of what allowed us to respond quickly were some of that basic work on viruses that weren't causing problems, or the Human Genome Project, or the technologies that came out of that. And then finally, I think, again, the tools are out there, and I think that the pandemic has really shown us is that the tools are out there to make infectious disease far less burdensome, not only here, but globally. And I don't think there's, at this point, we've shown what our technology and our responses can be, and I don't see any reason to accept the status quo anymore on this front. Okay, so I'm going to stop there. Thank you for coming, and I'm happy to take any questions. Ryan.
So a big part of it is also duration of the response, right? How long you're. So my question is mainly about duration of response. And so is there anything we can do to engineer the vaccines to have a longer duration of response instead of kind of these episodic, constant boosting and things like that?
Yeah, that's a great question. I mean, so even if you were to make antibodies for a few weeks that were great, and then then they sort of fell off, that sort of defeats the purpose. And so I think here again we can learn some lessons from other vaccines that do great. So for instance, the HPV vaccine is amazing. So after you complete the primary series of the human papillomavirus vaccine, those antibodies are maintained forever, for as long as you live. And so I think there's some features of the vaccines that work really well and give you lifelong immunity relative to the ones that don't so much. And I think one of the key features is that you generate structures that look like the virus. And so you generate these lattices and these antigen densities that resemble a virus more so than if you just immunize with a given protein, for instance. And so it's one of the reasons why I was actually really excited about the Novovax vaccine, because it sort of meets some of those criteria. So the good thing is about those pan-Sarbeca vaccines or the pan-coronavirus vaccines is that they all adopt that particular technology. And I think it's one of the areas where I think the mRNA vaccines could actually get a little bit better. The way it works right now is it puts up a spike protein on the surface of a 10 micron cell instead of putting up 25 of them in 100 nanometer diameter. And that makes a big difference in terms of how well the immune system sees it and how long antibodies are produced for. So the question is it compatible with the current mRNA technology? It is, but you have to include these sequences that allow the spike protein to then have the cells self-assemble them into these little nanoparticles. So that is actually one of the platforms, and that is one of the, I think, less dramatic tweaks you could make to the mRNA vaccine so that they might work a little bit more durably.
I have a question from someone online who asks, what can be done if a COVID-19 vaccine or booster results in a person's own healthy platelets being attacked? I know that's a rare occurrence.
Yeah, that's not, so there's some rare things that happen with the adenovirus vaccine. So that's J&J and the AstraZeneca vaccines. And so the risk, and so for these reasons, the FDA has actually gotten to the point where they're not recommending those particular vaccines, which is a little bit of a shame on one hand, because they have some features that are actually things we could learn some things from. So getting back to Ryan Sprisler's question right now, the J&J vaccine is actually very durable. It doesn't give you the same levels of antibodies that the mRNA vaccines do, and so therefore they're not quite as efficacious, but it's very persistent. But those rare side effects, there's some, depending on the demographic. And so usually the risk is highest in women under the age of 50. And so that risk can be somewhere in the vicinity of about 1 in 100,000. And it's serious enough where the FDA has recommended against the use of those vaccines. The mRNA vaccines don't do that. And so I don't think that's a particular issue. I think there are some things, and this is getting a little out of my wheelhouse, but I think, for those rare cases, I think that physicians have been made aware of it, and there are some things you can do specifically, so long as they're made aware that you received the vaccine a couple weeks ago. Mike.
I've got a whole bunch of questions, but I'll just give you one.
All right.
What's the current state of what we know and don't know about long COVID?
Yeah. What do we know? I mean, I think it's more that we don't know, I would say. So the question is, what causes it? I think that one of the leading hypotheses for which there's at least not half bad data is that for some people, for some reason, the virus persists. And so that even though it can't really be detected by the normal methods, you can actually see it in intestinal biopsies. And for some people, for a smaller group of people, by some estimates, like, something like 10% maybe even of long COVID sufferers are anecdotally reporting benefit by taking Paxlovit. And so the implication then is that there's some latent hidden viral reservoir, which is actually the source of the problem. That's probably a pretty small fraction of the overall long COVID. And it's really diverse because it can be anything from people who haven't fully recovered their sense of smell and taste, which is by far the most common form of long COVID, to other things that are a little bit more serious. One of the real challenges, I think, is trying to estimate the actual frequency by which long COVID happens, and the estimates vary wildly depending upon the definition and whether or not it's being compared to people who recover from other infections or for people who have had no infections. And so, the estimates vary anywhere from 1% to 50% in terms of people who end up with long COVID, and I don't really know where the actual number lies.
In your area of study of flaviviruses, is there anything from COVID-19 that applies in that area for you? And second part of the question is, if you had a candidate within the flaviviruses that could really cause a problem of a global scale, what would you choose and why?
So from the COVID, so we answer the first part first. Absolutely, and I think that the structure-based vaccinology, I think, is the biggest lesson. I mean, I would say that there are lots of people that were working on that anyway, including our own lab, before the COVID-19 pandemic. But I think it just really showed what had the power of that particular approach to make sure that you can control the immune system and what it's aimed at. The mRNA vaccines are actually a pretty interesting platform because there actually have been some early preclinical stuff on Zika virus vaccines with the mRNA. Moderna actually had some preclinical studies that they published on that. It just never really moved it forward, but now I think it's probably back online. And so I think some of the technologies that came from COVID-19 can absolutely be applied to the flaviviruses as well. If I had to pick one, I try not to make predictions because I'm usually wrong on this. I mean, I think what I will say is that no one would have predicted Zika, no one. Zika's been with us for decades and it never really has caused any problems. But, it seems like probably what happened is that it picked up a couple of mutations that allowed it to persist in mosquitoes a little bit better. And then that's really what the trigger was of the whole pandemic, just a couple of little changes in ways that you wouldn't have expected. So, we study West Nile virus. I have no idea if that's going to be, that virus in particular is going to be the source of the next pandemic. But I will say that last summer was the worst West Nile virus set of outbreaks that we've had in the state of Arizona in documented history, right? So I don't know if that's because it rained a lot, or if that's something that's coming, down the road. But again, it's one of these things where it's better to be ready ahead of time.
Do you know if any of the mRNA vaccines are being targeted towards influenza, since the current influenza vaccines have widely varying efficacy year to year?
Yes, they are. And I know that at least Mrna is planning to roll something out in the fall. And they've done some what are called non-inferiority tests versus the standard seasonal influenza vaccines, and it's certainly not worse than what those are. I think the real advantage, but I would also say that it's not as dramatically better as we would have hoped. So I think that the real advantage of those mRNA vaccines for influenza is that you can wait a little bit longer to be sure that you're matching against the circulating strains, because with some frequency, we get that wrong. So that if you can wait a couple of months before you actually develop, and the mRNA vaccines are so easy to manufacture, that could actually improve the efficacy just from that.
In an earlier lecture from the SEAL group, there was some discussion about efficacy of monoclonal antibodies against the current versions of Omicron and Delta and et cetera, and how the antibodies are basically much less effective in treatment of COVID. What implications might that have for the research that you do?
Yeah, I mean, I think what we've known for a long time is that antibodies work the best if they're already there ahead of time. Once the virus is already in the cells and replicating and spreading across cells, then you really need more of a coordinated response from the immune system. But if those antibodies are already pre-existing, then they can actually prevent the infection from ever occurring in the first place. And so, we've known this for a long time, is that the earlier you give those monoclonal antibodies, the better that they work. What happens in COVID is that there's a phase where you're sick because you have a lot of virus. And then there's the next and more dangerous phase where your immune system starts to misfire and cause damage to your lungs. And if you are at that phase, then that's beyond the point in which a monoclonal antibody can still help you. And so that's sort of the basis of the issues with the monoclonal antibodies. What I'll also say is that the monoclonal antibodies, as opposed to vaccines, they're aiming at one thing. And so it ends up being a lot more likely that a virus will be able to mutate its way out of that than when you have antibodies that are basically targeting all different parts of that spike protein. Okay. All right, if there are no more questions, thank you so much for coming.